All right, I don't know where Simon is, but we'll start. So, who wasn't at the genome assembly tutorial this morning? Yep. All right, that's all right. I'm quickly going to recap what we did. So, for those who just came in a bit late, this is the Genome Annotation tutorial. If you're in the wrong one, feel free to leave now. This is the Maple room. Everything we do is sort of linked off from this tiny URL, tinyurl.com gcc16-annotation. So that page is kind of where you should be if you get lost or need to go back. This is sort of our agenda for the day. So first going to just make sure everybody's at the URL, which you're doing now. I'm going to do a talk on bacterial genome annotation, some slides. Then we're going to actually do a genome annotation in Galaxy using Procker and then visualize it with JBrowse. And we're going to assemble the genome. So we're going to annotate the 6-contig genome we assembled in the previous tube. You don't have to reassemble it. There's another data set that's in the data set. You can just assemble that. If you're keen though, you can annotate whatever genome you want. If you happen to have a FASTA file from one of your projects at work or whatever, feel free to annotate that instead. I think it'll still work, but you can still probably go through with the actual tutorial. And then we have a speaker sitting at the back who's going to talk about WebApollo, which is an annotation curation system. And James is preparing it all now. Oh, yes, all good. All right. So yeah, I'll be speaking first. Annette's over here. Annette will be a helper. Simon's looking for a toilet that isn't clogged up like the ones around there. The male toilets aren't working, so you'll have to go elsewhere if you need to go to the loo. If I forget to give you a break, feel free to put your hand up and say, please let us have a break. And you can interrupt any time with questions. It's very casual. So for those of you who won't think, you may notice we're from Australia. Does anyone notice anything different about this slide compared to the last session? Added a snake. Good, you were paying attention. Is that still readable with the bright light? Or do you prefer? Did you know what type of? What type of snake is it? Is it a brown snake? I'm not sure. It was meant to be a brown snake, but it doesn't look particularly like one. What's the second thing? Does anyone know what the spider is? Redback, yeah. Oh. Yeah. All right. So just a quick recap. Here, this is about small genome annotation, and in fact, it's specifically going to be about bacterial genome annotation. We can sort of do viruses and other things, but we're focusing on bacteria. Just a reminder of sort of the relative sizes of bacterial genomes are quite small compared to, say, a human genome, but they still have quite a lot of genes. And what are we actually annotating? Well, we're annotating a genome, but do we have an actual finished genome? No, we usually have contigs that resulted from the sort of sequencing process and the assembly process. We end up with a bunch of contiguous sequences called contigs, and together they represent sort of most of the original genome that we sequenced. So typically you're going to get this, a file with lots of contigs. If you're lucky, you'll get something on the right. Today we're going to assemble a sort of something in between, a 6-contig segment of a bacterial genome. But like I said, if you've got your own genome or you want to download one, feel free to just use that instead. So what is annotation? What it really is adding biological information to sequences. The contigs you get out are just strings of AGTC. By themselves, they're not that meaningful. We want to actually know what's in there, right? So we can sort of use it to explain things. So here's an example of a segment of a genome where I've basically annotated bits of it. So the big green blob there is a protein coding gene. So it's got a start codon, the ATG. It's got a stop codon, TAG. And it's been annotated there, that green little sticky note. So it's been annotated as, say, delta toxin. So that's an example of an annotation. It's got a PubMed identifier associated with it in case you wanted to find out more about the delta toxin. That's kind of an annotation. This here is a ribosome binding site. So that's where the ribosome will attach to for on the transcript that comes from this region of the genome. So that's a small little feature that's been annotated. Annotation is basically anything. It doesn't actually have to be a genetic feature per se or something with biological relevance. For example, this homopolar run of T, it may have no biological significance at all, but it's kind of interesting because maybe we don't know what it means yet. That's still an annotation. So any labeling of your sequence is an annotation. So what's the sort of minimum information you need for an annotation? Well, obviously you need to know what sequence it came from. In our case, we've got a genome assembly with lots of contigs. We need to know the ID of the contig. The next thing we need to know is where on the sequence is it? And usually we do this with coordinates. So begin and end coordinate. Usually those coordinates are inclusive. They're included in the feature includes those positions. Has anyone ever used bed files? Yeah, so they use a different kind of coordinate system. It's not one-based. It's quite, yeah, so some different coordinate systems are used, but this usually, here I'm talking about sort of one-based where position 100 is in the annotation. And then the strand. So because DNA has two complementary strands, most features will have a strand associated with them. They don't have to. Some features are sort of independent of the strand. They just literally mean that locus of the double strand of DNA. But usually there might be a strand that it applies to, so the forward strand or the reverse strand. So that kind of gives us a position on a sequence in our assembly. Now, the next kind of thing we normally want, the main kind of feature label that we have is called the feature type usually. So there's all sorts of feature types. So back here, we saw a few different ones. We saw this is a coding sequence. This is a coding region that becomes a protein. This is a tRNA. This is a tandem repeat. And the ribosome binding site. These are all sort of feature types. These are sort of biologically useful things. So there can be a whole hierarchy of feature types or an ontology. It can be flat sort of hierarchy thing. I'm not going to go into that too much, but the idea is that features have a type and then they have kind of attributes. So protein coding is the most common kind of feature type in a bacterial genome. But then you'll have all these attributes. So it's a protein coding gene, but what is the protein that it makes? What actually is it? So there's all sorts of proteins that a genome will have. Example there is an alcohol dehydrogenase. It might have another attribute, this enzyme code. So that has an enzyme code of 1111. Subcellular location might be something like where does the protein live in the cell? So in this case, it's the cytoplasm. It can be notes. It can be anything. So if you ever look at an annotated GenBank file or something, you'll see all sorts of attributes that could be annotated. So I'll just go through some of the sort of the common bacterial ones. So I guess you can broadly split the feature types you'll encounter in bacterial annotations into sort of protein coding genes, which the bulk of the genome is made up of, and then non-coding genes. So non-coding genes include sort of things that are transcribed but not translated. So I guess I don't really have a feel for how much molecular biology people understand. It might be quite variable in the audience, but doesn't hurt. But I think I might not have enough detail. I'm not sure. So there's a bunch of non-coding things, and there's protein coding genes. And protein coding genes are really made up of a bunch of sub-features, which I'll show you here. So this is kind of important to understand this protein coding feature because it is the bulk of your genome. And this is the bulk of the things when we're annotating is what we're trying to label and correctly annotate. So this is a canonical bacterial gene. So the red line there is the sort of the genome. And there's a promoter, which is where the polymerase, and if there's proper biologists in the audience, you can correct me if I'm saying it wrong, where the polymerase attaches to the promoter, and it goes along, then it hits the orange bit here, and starts transcribing to the end of the red bit, to RNA. That goes from the DNA to RNA. So then we've got this RNA molecule here underneath, which is floating around. And then the ribosome comes along. It attaches to that ribosome binding site here. Then it sees a start codon. Then it starts translating to protein until it hits the stop codon. And now we've got a protein sequence, which then folds up and does its thing. So how hard is it to find these in a genome sequence? Given just a genome sequence, how can you find them? I forgot to put a slide on open reading frames, which is really silly. Do people have questions about this? Like, I would. Do people understand genetic structure? I'm not sure. Okay. All right. I'll just go on. We could annotate this by, loading up a genome sequence into a favorite genome viewer, IGV, Artemis, whatever, and actually going and finding them, creating a feature, labeling it. But obviously we want to automate this, or at least to bootstrap the whole process. So things like tRNAs, tRNAs are the sort of part of the machinery that brings amino acids into the ribosome to make proteins. And they're quite easy to find. They have a kind of a well-established sort of conserved structure, and they've got a variable bit in it to do with the codon that they represent. Ribosomal RNAs are pretty easy to find as well. Lots of software. They're highly conserved in bacteria, so they're easy to find. Coding regions, as I said, are the sort of the bulk of the annotation. So they're pretty easy to find because they're large open reading frames. That's stretches of the DNA that have no stop codons in it. And if you load up a genome, which I don't have here, you can just see them. They just stand out. So they're quite easy to find the open reading frame. The hard part is figuring out where the start codon is. So eukaryotics only have one start codon, which is a methionine. So an ATG is a start codon. Bacteria can have up to 5 different start codons. Most bacteria will use ATG for 80% of the genes, but then there's another 2 start codons that they will use less frequently, and then there's even another 2 that some bacteria will use. So sometimes you don't identify the correct start codon, but it's better to find your gene and have the wrong start codon than to not find it at all. So the other problems we have is that when we have genome assemblies that are not complete, you've got, how we have to break our contigs. Our contigs get broken into pieces then you can have genes that are broken. So you might not be able to find a completely intact gene because of these contig breaks. Pseudogenes are genes that are broken for real, like in the organism, they are broken. They're not intact. They're in 2 pieces, 3 pieces. That's just sort of from reductive evolution or some event that's happened that didn't confer a selective disadvantage, so it just stayed broken. So sometimes it's difficult to figure out maybe assembly errors from actually real pseudogenes. That's often a problem. So finding them isn't that hard. Figuring out what they potentially do, what their function is in the organism is probably where most of the computation goes. So there's kind of 2 strategies for identifying these coding regions. So the first one, I'll just start here actually, is with what I talked about. You can just ab initio gene find. It's like de novo genome assembly. It's where you don't have reference to anything else. What you can do is you can just go through your genome looking for open reading frames, which are areas of the genome with no stop codons. And you might have a model of what ribosome binding sites are also a little bit conserved, so you can sort of figure out where they are. Therefore, you can predict where the start codon might be. And there you go. You've predicted a gene. Another way to do it is to not look at open reading frames specifically, but to take all known protein sequences and align them to your genome. And if they fully align and they align perfectly or no gaps, more to the point, then you can just sort of say, okay, that region of the genome is that protein. But the problem with this is that you will miss things that if there's some novel proteins in your organism and they're not in the database, you won't be able to find them. And you may miss partial proteins. So typically, you might use a combination of these things. So there's software out there that kind of does this called Maker that Peter before mentioned when we were at the start of the lesson. That will try and do both of these things. So yeah, there is a bunch of software for bacterial annotation. Has anyone heard of RAST or MG-RAST if they've done metagenomes? So RAST is a sort of an online gene annotation system. It kind of uses both approaches. It'll find novel genes and it'll also align existing genes. XBase only does ab initio finding, and I don't think it's actually running anymore. This is a standalone tool that doesn't use ab initio at all. It actually only aligns, and it takes a long time to run. It isn't really being used. And this is, if you've ever submitted a genome to NCBI, they can do the annotation for you. It's really actually quite a good annotator, but you can't just like go to a web page and upload it. You have to submit and then wait 3 months for them to annotate it. It's very frustrating. So they won't release the software either because it's highly tied to the internal infrastructure at NCBI. So I got very annoyed at that. And so that's why I wrote Procker. So Procker is a sort of a command line tool that's wrapped in Galaxy. And I wrote it because I got frustrated with all these tools. So I wanted it to be as simple as possible. You just give it a sequence and it will give you an answer, right? And I wanted it to be fast so I could go get my coffee and come back and it was done. And I wanted, now it's too fast. I don't have enough time to get my coffee. It's standards compliant, so it outputs like these standardized annotation file formats like GFF and GenBank. Yeah, and I tried to make it as sort of consistent and trustworthy as possible. And I'll go through how that sort of works. Well, that's, I guess, I say Procker, but I notice most people pronounce it Procker. It's obviously just a sort of an accent cultural thing. So, yeah, that's my motivation for calling it that. Has anyone ever seen a quokka? Yeah, I haven't seen one either. So they live on an island off. Perth is on the west side of Australia, and they live on an island off the coast. So, have you seen one, Simon? Yeah. Oh, all right, something. Are they cute? Yeah. But yeah, so it's a giant rat basically. So they live on an island called Rottnest Island, and it was discovered by the Dutch. Rottnest means rat's nest because they look like rats. Yeah. This is Procker. This is internally what Procker is doing. Everyone knows what a FASTA file is. It's just a file with our contigs. So you produce that when you do the genome assembly. And it's basically a simple pipeline. So it uses a suite of existing tools, like no point reinventing the wheel when there's good stuff out there. It uses different tools to predict different types of features, and then it kind of assimilates them all. I'll go in a bit more detail about sort of how some of these steps work. So yeah, what can you trust? So has anyone ever looked at a genome annotation, used a genome annotation off GenBank in their work, or any of your collaborators? Well, if you do, you'll start to realize that there's lots of mistakes in there. And I didn't want to contribute to that propagation of errors throughout GenBank. So I guess why are all these mistakes in GenBank? So we predicted this gene, and ideally we want to know what its function is, right? But when I say function, I mean, what's its biological function? What's it doing? What pathways does it participate in? What physical, what does it do? So not just like what's its sequence. So we don't really know how to do that. We can't just look at a protein sequence and figure out what it does, right? So how we do it is kind of use sequence similarity to sort of figure out what its function is. And because we have proteins that we know what they do, they've been characterized. And so if our sequence is pretty much similar to that, well, we'll just assume that it's doing the same thing. Most of the time it's probably right because, you know, all these things had common ancestors, and a lot of these gene structures are conserved. Even there's genes between human and bacteria that are conserved. So using sequence similarity is our main kind of tool that we can use to try and figure out what our proteins are doing by basing them on what we know about other proteins that do have real evidence. So there's two ways to sort of measure sequence similarity. One of them is sort of using the sequence, so like directly comparing the sequences within the database of sequences, like at the sort of nucleotide level. So probably most people know what BLAST is, I would hope. So the typical thing you would do is get your protein and blast it against the database, and then if it says it's something kinase, and we'll just say ours is a something kinase. So that's fine. There's more sensitive ways to search things where instead of searching for specific sequences of patterns, we sort of build these models. There's like this position-specific scoring model, matrices, there's a hidden Markov model. I'm not going to go into details. There's lots of different tools and databases where people have built protein families, things like PFAM and stuff like that. PFAM down there and PAGFAM. So these are kind of more sensitive but slower. These are faster but less sensitive. I guess this slide's to talk about what a lot of people do. So the non-redundant database is kind of a database of all the proteins that are in GenBank. So a lot of people think, oh, that's got everything in it. Why don't I just search all my sequences against it, right? And just that's the best thing I could do. The problem is everything's in there, and everything means all the junk and all the rubbish that people incorrectly annotated. So yeah, it's not very picky. So if you put garbage in, if you put garbage into your annotation pipeline, what you get out is going to have garbage in it. And also it's big, so it's going to take ages to search. So I guess the way Procker gets its speed is sort of because of these facts. So it's mostly true that searching against a smaller database is fast, and searching against things that are similar to you is fast. So Procker sort of has a multi-pass, attacks the problem sort of with a hierarchical multi-pass thing. So it starts with a highly curated set of proteins, which is kind of this core bacterial proteome that I get from UniProt. And then it sort of advances to more distant databases as you go along to sort of speed it up. And this seems to work pretty well. So what is this core set of proteins I annotate? What I do is I use UniProt. So UniProt is a protein database. Very well annotated, and most importantly, there's annotations in there to do with evidence. So each entry in this database says why it's in there, whether it's been experimentally validated. Like, did someone actually see do a proteomics experiment and see this protein, or they have levels of evidence to do with mRNA. Did they see a transcript in a transcriptome, like actual RNA evidence that produced this protein? And so what we do is I basically take this giant database and shrink it down to things that are only curated, only bacterial, only have strong experimental evidence for them, aren't fragmented. And then I create this mini database, and this basically has kind of 50% of the genes, trustworthy genes, of any bacteria. And I just use, BLAST gets to sort of achieve that very quickly. And this kind of solves half the problem very quickly, and then we can move on to the more slower methods. So these are basically the slower methods. So you can create your own databases for your genus. If you're only working on TB, you can create a tuberculosis database with just TB proteins in it. And this kind of makes sure that you pick up on the naming convention of each discipline. So although there's proteins that are conserved across like all these different species, different species people, they call the same protein different things. It's like they have their own language for that set of genes. It's normally a historical thing, but if you give them a generic name for their gene, they sometimes get a bit upset. So because they're so used to seeing their particular name. So you want to try and make it domain specific and might create a custom database to use. We go through all this stuff. If we don't find anything we can trust, we just call it a hypothetical protein. So provenance. Provenance is kind of this idea of recording where an annotation came from. So as I said, there's a lot of stuff in GenBank that's been called something, but we don't really know why it got called that. Where did they get the idea for calling it that? What was the evidence for it? So GenBank and these have a way to sort of record this sort of evidence information, this thing. So basically, they've got some tags. Now these tags change regularly, unfortunately, but there's tags for sort of wet lab evidence and tags for sort of dry lab evidence. So Procker will insert these tags in your annotation so that there's a record of how it came up with it. So I think this is pretty important. So it will tell you how it, what software and what version they used to predict the gene, say, how it came up with the protein name or whatever. So I think that's one of the key things that I think, well, one of the important things I think it does. You don't always need to refer it, but if you ever do, you'll know why it came up with that. This is just an example sort of of a tRNA feature type, what it would look like. Remember, features have a type and a location and then a bunch of attributes. So this is sort of the GenBank ontology, or the GenBank language for is a sort of a fixed vocabulary of things that you can use for describing tRNA types. Like it's got a gene attribute. The anticodon is where in the sequence the anticodon was for the tRNA, and it's used for leucine, amino acids, and so on. And it was predicted. This is what Proctor puts in this inference. It says what's the software used was used to predict it. So people are using Proctor. The Sanger Institute is using it. So they've annotated 100,000 genomes with it so far, which is pretty impressive. And they told me originally that they did 50,000 genomes in 2 weeks, which they calculated it took 24 seconds a genome to annotate, which they must have a very good cluster because I can't reproduce that. No, I don't have slides on each subtool. Are you? So I think what we'll do now is we're going to just do the shoot. I think we're going to get a feel for what and look at an annotation. So if we go back to the program, has everybody gone to this URL here? Put your hand up if you still need me to have it on the screen. Yep, we'll wait. Because
rearranges the order of
it needs to be in the correct format for submission to GenBank and have the right file extension. And also for completion's sake, the folder it creates as a bundle you can give. Yeah, I wish it was that easy.
Well, but I mean, yeah, I noticed,
Collaborate.
I noticed the naming convention is the same as GenBank.
Yeah. So, all right, you've got the URL. Yeah, go.
Going back while we're waiting.
I'm sure you.
I know people are still maintaining PFAM, but microfam, is anybody still thinking that?
Not that I know of. It seems to change occasionally, but it's no one really maintaining. But it's still actually quite good for bacterial protein families. There's definitely families in there that aren't in PFAM. So whether we can trust them, I don't know.
Well, it's mostly because of the
Yeah.
what's out there.
I don't actually use it anymore in my own work. It's mainly because I can't distribute. It's too big to distribute. It's quite painful to distribute my software because of the sizes. You can't put the stuff in GitHub and stuff like that because they're too big. Sorry?
So what's too big?
Some of these extra databases that Proctor searches against. You can give it as many databases as you want to search against, but these databases are big. Some of these Hammer HMM ones. So
Sorry, just a side. And I mean, this is something that the people in particle physics started. There's a thing called CVMFS.
CV, what?
CVMFS. It's basically a CERN-backed fuse-orange database that you can mount a direction and it goes out onto the internet and finds a data store. And then as soon as you touch the file, it downloads and caches it. It's basically for read-only data and beautiful for reference data sets.
It sounds good, but it's not helping Joe Blow on his MacBook who's annotating a gene.
So if you're running on PC, well, I don't know. If you're doing bioinformatics on your MacBook, it's not going to help. Okay? But if you have a small server, things like that, I'm getting late to write up how they use it. So that, for instance, the reference data collection from use data that all is available on CVMFS. You have the whole reference data collection that they have. but the same thing with the kind of basic.
Okay,
That's the kind of thing that we should be providing.
well, talk to Simon. So we're going to, no, it sounds pretty slick. That's a nice.
Very slick. I mean, the Prerequisites guys, they come out kind of nice in terms of installing size. I think it's quite easy.
All right, we're going to do the tut now. So the tutorial instructions are off this page here. You've got to load from the shared history. It tells you how to load from the shared history of the data set. And basically look for the spades contigs, which I hope are in there. And we're going to assemble those. But like I said, if you want to annotate something else, go for it.
There's two servers. We sort of do half and half.
Have you updated the?
Have you fixed the JBrowse on the second server?
Yes.
Because it wasn't 60 minutes ago.
Now, but it's in NGS assembly instead.
Choose one of those servers to use. If you're at the previous tut, you won't need to register again. But if you weren't, or you're changing server, you'll have to register again. We won't use your email for anything. This is a temporary cloud instance, which will
Go away after about a month.
be destroyed in a month. So, yeah. We want you to try and alternate between those servers so that you don't have to wait too long for your annotation.
How do you possibly alternate? Last name begins with...
No, I'm not going to constrain anything. People can choose one. I'm going to go with
Random. So, well, just tell them what each of those subprograms actually do.
maybe after the shoot.
Yeah.
I don't no time. I don't think it's useful, to be honest. Yeah. I don't know. How are you going to do it?
I think protocol's important.
Yeah.
And Aragon.
Really? I don't even know how to tell me.
Infernal.
You know how that works?
Kind of.
I think we just get this JBrowse. This will take a while, I think, this tut for the JBrowse bit maybe.
Maybe.
Sorry. The one seed to use, one. Oh, it's only a small genome. So the genome we're annotating is only the first 150 little base.
Yes, space assembly, all that.
Yeah, just put your hand up if you're having trouble.
So the kids may not be so
You need to register on the server that you can use on the top. Yeah, and then register. Doesn't really matter. Yeah. Just a simple keep when there's yeah, you guys.
I have
was reading this different work on this morning.
Usually it's only one. It's only one scaffold. It's usually only one scaffold. Scaffold has gaps.
Data subscriptions for 3A.
Most people probably use scaffold. We'll step right.
Oh, it's
No, you don't have to. That's just for labeling mostly. That's in there. There it potentially could be used, not quite, but I sometimes have some databases with rocket specific genus.
Everyone's trying to use it at once.
If you do specify it,
So you got
it might,
four
there's an option for use the genus and then use a special transformation.
Yeah.
Doesn't gain you much advantage. Something broken?
Stay silent.
What?
I'm looking at the Spades manual. I can't find, what I can find is references to doing automatic payment choice in the future.
Right.
What do you mean?
Automatic payment choices in Spades.
You just don't specify.
They just leave it out. And then manual just, okay, so then,
It uses
this is the wrapper. There you go. Just change that.
Oh, it doesn't necessarily choose good changes.
No, it is. I'm just saying that, like, you want that as an option.
Yeah, so you just don't put the K value, you leave it off completely. You have to kind of do a double wrapper option.
This is a full quote. Run. Maybe. Yeah. Yes, right. Oh, yeah, but that's the around. Okay.
So don't blame me, I didn't do the interface for this tool, but this one's a little bit old now, but we are going to redo the interface to make it a bit simpler. It's a bit complicated at the moment. We're just going to hide everything behind an advanced tag and just have the minimum stuff. So you'll notice that Procker produces a whole lot of files in the output. Some of the important ones is the log, so it tells you what it's actually done. And so you can see this one here has got gone through and found some tRNAs and it's found some rRNAs, and then it's gone through and tried to saying that it's using Hummer to find some bits and pieces and trying to prescribe function to different things. It's fixed up some duplicate genes and then produces a whole bunch of applet files.
So yeah, when you run this on the command line, it's fun to watch it and go through all the things. It's like meditation.
Yes, but
What if we got an error that says, please rename your proteins?
You didn't follow the instructions exactly correctly.
Oh, yeah, I didn't show you the tiny URL.
oh yeah,
I don't have the instructions.
tinyurl.com,
there it is at the top of the page there.
GCCs, yeah. Yes, the bane of my existence is that error that you just got. And this error comes about.
A few other people got it too.
Well, then that means they didn't follow the instructions exactly correctly, because it tells you to change a couple of things to single letters. If you don't do it, you'll get the error. The problem stems from Spades and Velvet and all these software. They produce config names that are really long. You know, they've got the config name underscore coverage underscore length underscore number. And GenBank has a limitation of 37 characters for a config name. And so we need to tell Procker to sort of, the Procker on Galaxy is unfortunately too old as well. I've resolved some of these problems. It's way too old. It's 2 or 3 years old.
3 years old.
We need to think to address that pretty soon.
So in the .text file is a summary of what Procker found. So it'll says it started off with 6 contigs. It's found 151 genes. 149 of those were coding regions, and there were 2 tRNAs. That's pretty cool.
This is only a partial genome, so that's why not everything's there.
Well, the error one, we won't worry about
That's,
it.
oh yeah, we're going through each of the files. That's good.
Yeah. Do you want to talk about the error one, discrepancy report? Maybe.
So this is NCBI. They provide a tool that gives you this error report. Most of it's indecipherable. Yeah, don't email me about it, please.
But the important ones for us are going to be the GenBank file, this one here, which says that on contig 1, it found a gene, and it's called gene 1, and it is an integrase core domain protein. There you go. That's probably not that surprising.
That annotation, is that taken from EFAN?
Where did it come from? If only we had inference tags to tell us how we came up with it.
Yeah, came from PFAM.
So Prodigal was the predictor for the gene coordinates, and it was from the PFAM database from a match to that PFAM accession number. So you can exactly tell where I got it from.
Cool.
So that you get like a GenBank entry for every config. So we've got a lot of small contigs here. Maybe if you scroll right to the bottom, that's the big config, I believe.
Well, this one will be contig 3.
So this config's got more than one gene in it.
Has it got any named genes?
A replication protein and then another replication protein. That's a bit suspicious. Oh, plasmid recombination enzyme.
So with bacterial application, how reliable is the detection of the UTR?
There is no prediction of UTR. We only predict the coding region. There are no software that predicts the UTR.
Yeah, I mean, that's
So this one might be a bit interesting if you found a brown myosin resistance protein.
in bacteria, does it make sense to use gene ontology annotation?
Good question. Gene ontology. Anyone in the audience want to comment on gene ontology? Oh, you in the back. This audience member knows a bit about gene ontology.
Yeah, we could. I mean,
Yeah, so
I was going to do it offline and just why not. I will say that for prokaryotes and microbials, we're weak. We just haven't had groups to work with to help us other than people who were originally E. coli, but still we're quite weak as far as the terms that are there. Viruses are okay on pretty good, but it takes a group to work with us to say, here's what's missing. I would like to see it.
Yeah. So it's one of my most often requested features is to add. So Go ontologies, it's like a control vocabulary for describing function and molecular stuff about proteins. So as they're not as good. They used a lot of eukaryotics because they've been well curated. Bacteria less so, but they exist. And so I could put Go annotations in because I infer stuff from Uniprot. Uniprot has Go annotations associated with each of these. I just haven't integrated the logic into Proctor.
Interpro too.
I don't use Interpro.
Oh, okay.
Otherwise, I'd be here all week just running some scanning against Interpro would take me, take the whole day, half a day for bacteria.
Okay.
I needed something that would run on people's desktop. That was the niche. They really needed something they could do on the desktop. So, but you're right. I could make Proctor 2's on the cards. Always version 2. PFAM has Go annotations as well.
That's where we got.
Yeah, so Proctor could do it. It's just the infrastructure isn't quite there yet, and I haven't figured out a nice.
That's working.
Okay.
File a GitHub issue.
Yes.
So anyway, this this GenBank file is kind of human readable. You can kind of scroll through it and get some detail about the things that were found on your sequence. But the one that we generally use downstream from here is this file, which is the GFF file. And basically, this is it's a file that contains it's more succinct than a GenBank file, but it contains the same information basically. And what it says is that each line in a GenBank file is one feature. So we can see here this one for Contig 3, Proctor found a gene at position 336 to 1109. If we scroll across, it shows all the information about the inferences, what its product is, etc., but it's less human readable.
It's very machine readable.
Yeah, very, very, very machine readable.
So it's tab separated, so you could load it into Excel, basically, and get most of the columns. This last one is a bit more detailed. Whereas the previous, you just go back to the GenBank file. This is an old format. I wouldn't know what year the first GenBank file was ever used, but it's designed to be, I think it's like 60 columns wide. So for the more mature, mature people in the audience, that was so it could be printed on a 80-column dot matrix printer, or that doesn't really matter today. So that's why it's kind of got this fixed width, whereas GFF doesn't have any of those.
It used to come out on floppies, didn't it?
And the main problem is that there are some protein names that are longer that you can fit in this column in 60 characters. There's some protein names like phospho something, blah, 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 fructokinase, and it doesn't fit. And so you have to put a break in there, an artificial hyphen in there just to make it fit in this format. So a little limitations like that, historical limitations. Sorry, it was
Tell me what we're doing.
anecdote aside. Did the GFF.
All right, so then there's some other really useful things that Proctor produces. It has the FNA, which is the contigs, which are then in the same order as they are in the GBK and the GFF file. And then there's this one, the FAA file, which stands for fast A amino acid, and that's the all of the actual protein sequences of all the proteins that it found, which is kind of handy.
You could build a BLAST database from this, or what actually is quite useful for, if anyone comes to you and is doing a proteomics experiment on their genome, the Proteomics Center will ask for a file of proteins to match peptides against. This is what you could give them.
And then there's the FFN file, which is exactly the same as the FAA, but in nucleotide space rather than amino acid space. And then there's the ones that NCBI needs, like this sequin file and the FSA file and the table file and all those things.
These are sort of needed if you want to submit to NCBI.
GenBank, yeah.
Sorry, what was the difference between the FFN and the
Oh, so the FAA, so you see here it says,
FNA and FFM.
okay, so in
One
the
is the
FAA,
contigs, one is the original genomic contig.
Okay.
The other is just the coding sequences. That's a fair question.
Yeah.
These are sort of, they're not official extensions, but they're kind of
semi-standard.
semi-formal. NCBI uses them, so
Which
I just
is
got an issue that I don't produce FRN, which is all the RNA features. I'm not coding RNA.
So, I was just going to, no, we won't do that. I just want to show you what they look like when we build them. So,
Are you doing JBrowse?
yeah,
Did anyone get to the JBrowse part of the tutorial?
yeah,
Oh, cool.
awesome.
I had problems. One thing is where.
It's a slightly different version to what-
Wild type vegetable. I could not find wild type or how to add it
It's in a different history.
in the track data.
It's not on the second server.
And then when I.
What?
What's it?
No,
It's in the.
Oh, yeah, I'm on the one with the IP address.
Actually, it is in another.
it's in the other history. It's on that server.
Gotcha.
But,
Are you in the small genome annotation history? Did you
No,
like?
but the GFF is in the small genome assembly.
Yeah, it looks like that.
Uh,
yeah, but that's okay because,
You're not meant to use the wild type. You're meant to use the one.
We don't use one.
yeah, don't use the wild type.
Didn't match exactly.
Okay, fair enough. Well, I made it simpler on this one. I didn't put the wild type in there to confuse it.
Yeah, and then when I hit execute and got it up on the side, then it was.
Oh, it's not, it hasn't been taken off the whitelist yet. Okay.
Yeah, that was the thing.
That's my fault.
Should I go to the other?
No, it's okay. I'll just get it off the whitelist.
Simon, like, just in the break between the previous one and this one, has been setting up this second server to help cope with the load.
I'm a big breaker.
We're
That's
just
good.
too popular, that's the problem.
I knew I forgot to do something.
This is one problem with the cloud. So all these are sort of dynamically created on the cloud, these clusters we're doing here. And so it's not like you've got this nice server that everything's set up and always working. You kind of sometimes little things get lost.
So anyway, if you view your JBrowse thing now, it should work.
Uh,
I have a question with the JBrowse. It shows all three reading frames.
Yep.
Right? And but how do you know which one it's referencing when you click on that segment?
Question.
Yeah. Can you hassle the JBrowse people next
we're
week
saying
at the
hard
GMod
one.
conference to fix that?
No, not this week.
Is it?
Straight after.
Yeah.
There's a classic option and stuff, there's a track, and that'll take it back. And I think.
So we want to see where the coding region is, which we want to see.
Yeah, which one of the reading
wanna
frames
see
is
the frame.
the one that's the coding one when you click and it says this is the protein product? Which one of the reading frames are they talking about?
It tells you in the annotation, but you don't know. I want to see it overlaid on the coding region, on the
Or
frame.
even just make
like
it as a
Artemis.
different color or something.
Don't worry, I've already, I'll write to.
Well, Eric's right there.
But my understanding is JBrowse is about to get released like massive.
Is that true?
That's what the.
JBrowse is about to be rewritten massively.
That's what it. That's what the.
So anyway.
We're adding a number of features.
So I guess this looks like a genome browser to most people. But the cool part about this genome browser is that all of these sub features, etc., were all found by us in about 30 seconds in Procker. And then suddenly in Galaxy, we can just have a look at them. So we can zoom in a bit further and, yeah. Tells you what,
Did you enable the product?
not yet, I will.
I thought it automatically did now.
Yeah, I thought so too, but I don't know what Eric did. Labels, products, a prod cut. There you go.
Prod cut, I think that was the problem. Is that
It's a typo. There you go. So suddenly we've got the, so you can see the products on there as well now.
And then I like, there's all sorts of other ontologies besides Go. I know there's COGs, which are a common, an old uncurated system, but COG has like 20 categories of genes, and a lot of people know COG coloring, so they would want to color their genes like COG colors and things like that. So you zoom into the reading frame so we can exhibit the problem. One more. So the proteins are down here, but you don't know which reading frame they're in. It'd be great if it like did a rectangle around the reading. You can see the feature.
Well, you can see this is the stop code on, so it's probably in this bottom one here. Probably.
And probably have a way to deal with that.
Speaking of which.
Yeah, what do you mean? You can visually see it?
Yeah, you can have it color the transcripts
By the frame.
by the
Yeah.
frame.
Oh, cool.
So that you see, yeah.
Oh, so you're just using the frame attribute of this to color it. You still don't see it overlaid on the text.
Right, you still don't see it.
But they're different colors.
It does make it a lot easier, and we don't use the phrase
See?
colors for the sequence.
No, that will drive you insane after a while, those colors.
Does me.
You can turn it off on the view menu somewhere. Colors. Maybe not.
No.
On the little, yeah, sorry, that's not.
That's buried in there.
Yeah, it's probably buried in there somewhere.
So how many JBrowse web follow people are here? I mean, we need to lock you all in a room until our demands are met.
You're not really.
Fun.
We'll provide you with snacks.
So, speaking of web follow, what were we gonna do now?
All right, so people are still doing the shoot, are they? Or the JBrowse takes a little while. Huh? Oh, what time is that shoot?
Now by default.
People can do it if they want.
Yeah.
I can do it.
Yeah, and then, yeah.
Pull up some slides.
I've got some slides there. Change that to
Tips, I guess.
Yeah, I wasn't sure if they were.
Yeah, that's it. In a second, I just wanted to wait until people.
Let's have to switch monitors.
You've got a picture in this thing.
So just scroll across the
Okay.
Change context.
Okay.
No. No, it's just, well, personally,
So I'll show you.
no, so, Justin and I work in bacterial and we write the because of the fact that and it's one variance
Yeah, before name.
there is one. Because of problems. So most of the things are by experiment. It's the easiest one. Those two. Some
you know, I'll show but I want to, and they can actually give you a bit of boilerplate to help do things, but it's still very, very important.
Yes, I have to do that right now. Yeah, I'd like to be able to. Yeah, well, I'm gonna get started with one box to show what's about our tools.
I just, it's.
Oh, yeah.
Hold on.
So really something.
Yeah, that's what I show up.
This is a perfect, yeah?
Well, then he showed.
So how would I, is there a way for me to hook up? Otherwise, most of it.
You may go like this. You may go HTML laptop when you're ready.
Oh,
So,
all right. Well, you just tell me. Oh, good.
so are you going to talk about this for me first?
Yeah, I've got one slide before James takes over.
Hey.
Nathan.
Oh, sorry, did I call you James?
So I've got to look up your name like 3 times.
Nathan Dunn, right?
Yeah.
I should know, I know your Gmail.
Yeah.
What's LBL stand for?
Lawrence Berkeley.
Oh, of course. So I'll just sort of move along a bit. You're welcome to keep going, but I just want to say, so we've done this annotation. It's like an automatic annotation. It's kind of mostly right, but it's not going to be exactly right. And in fact, our understanding of proteins changes over time. A protein that we knew nothing about this year, in a few years may have been characterized in E. coli, I say, and it might be applicable to our bacteria. So we want to update our annotation. So I guess the thing to remember that we've got this annotation, most of it's pretty much right, but there's going to be some things that have been annotated that actually aren't even there at all. Often small proteins that they kind of get predicted, but they're actually never, they're not real coding regions. There's missing stuff, so things that we didn't find that should be there. And also, I alluded before that often we get the gene right, but we may get the start codon wrong, or the product might not be quite right, or for our domain might be a bit special, or it might have a snip in it that makes it slightly different to what we thought. So the process of fixing these annotations is kind of called curation. And this is like manual effort. It's not easy. I've done lots of curation on bacterial genomes over my career. It takes a lot of work. But we always, when we say sequence a new species, we always put a lot of effort into curating it so that we've kind of got a very good reference for the rest of our work. But the best way to do it is to spread the load, right? Many hands make light work. So community curation is kind of where we are heading in bacterial space. And I think there's obviously already community curation in larger organisms because you simply just can't, one individual can't do it. So Nathan's going to talk about a piece of software called Web Apollo, which is derived from the original Apollo, yes, I guess, which was a Java app. This is a sort of web version of that. It uses JBrowse as its kind of main engine, I think. But he'll talk about that and demo it and how it can be used for community curation. All right, you're plugged in. We'll switch you over.
And just before you go on, the Galaxy community in general is working on a way of having, so we've got JBrowse in Galaxy at the moment, and so a few of them are working on making Web Apollo available as a Galaxy plugin as well.
Yeah, and I'll show a little bit of this, and I think we're going to do a talk and a poster about it as well. But I just wanted, this is a great segue, and I love having someone else explain the problems of automated curation or automated annotation. So this is a different organism, but the screen should look very similar to what you just looked at. We are viewing a JBrowse view of this sample organism called Volvox, but doesn't matter. And so so the next slide, we'd hope, next year you'll be presenting with Apollo or something within Galaxy. You click on the view, and then you could, basically it takes you into this alternate product here. So this is Apollo, and it is loading up. It's going to take a quick second. It's not quite cached. But it's basically that JBrowse view underneath it. Anyway, so it's this JBrowse view underneath It's just taking just a second to load up. And so we have a single annotation here. And I'll show you a different example, but mostly I just wanted to show you what it looks like currently in Apollo. There we go. So all of those genomes in JBrowse that you were just looking at exist in Apollo, but you have this extra piece up here, which is this annotation region. So if you go through all the work of using Troster's tool and his group's tool to build these functional annotations, you obviously can pull in additional evidence and then and edit these by basically going and saying, well, I think this has, doesn't quite match the annotation we want, so we're going to create a tRNA out of it. So I know that's what it is. So we have this piece of data. You can see it showing up in the side panel. I can view details about it, coding regions, so on and so forth. Having two side panels is probably not the best thing, so we're going to close that. And so once it's in there, there's different types of annotation. I'll cover that a little bit, but one, if we have additional evidence, we can change the basically the structural structure of these. We can do things like going in and deleting exons, basically doing all those type of structural annotations. But additionally, we can go, we can change the annotation type. So if we decide it was a pseudogene, it'll go through and change that. And I'll show a little bit more here. I mean, you can do all of that within Apollo as it stands now, and then go and export GFF3, FASTA, Chatto, so on and so forth. So this is, Volvox is not a very interesting sequence since it's not real. And I guess the other thing I would point out is we have the ability to go through, and this is a horrible interface, but the ability to add additional attributes to a current annotation, to individual transcripts. Let's add another one, as well as, there we go. So anyway, you can add multiple isoforms and so forth. So, and so this current version is, and we're gonna present this later tomorrow as well via, there's a Docker compose script that'll bring all of this stuff up. But I'm gonna actually go back and just cover a little bit of the slides that I will be covering tomorrow. A lot of this was already covered by Troster, but so the structural annotations, I showed you the functional annotations, and you can edit both of these, and they both are result in both GFF3 and FASTA annotations. And so, and again, we just, I think today decided we were going to do this, so apologize. But like,
Nathan did this short notice to me. I asked him yesterday if he would contribute to the session, so he's done pretty well. So
yes.
anything to show them.
Yeah, so, and I guess the point is, like Troster's made over and over, is you have, if you have data that's not quite clean coming in, you have the ability to go through and manually annotate it to fix it. And so this gives you ability to,
Are you trying to share your screen on the stream?
No, I'm going to do this presentation again later,
Okay.
so, or the longer presentation, so it'll be,
Okay.
that's fine if it's not.
Okay.
And allowing people to come in and view additional data tracks is helpful. And the other point about, as Troster covered earlier, the nice thing about manual annotation is you have large groups of people, and people tend to focus on aspects of a genome or a set of organisms in a particular genomic region. So it's very common, or probably more common, to do this using a genomic browser. So I'm going to go back into, since we're just doing a demo, and show you sort of some of the other tool or things that it can do. So this is Honeybee. So this is actual data. Not sure what that comment was, but.
Well, I was going to get on too, but you're running it on your own box.
Oh, sorry. Yeah. We have a demo server. If you go to Genome Architect is the name of it, you can actually go through, there's a demo and all sorts of things, so you can mess around with it. And all the details I'm missing will be there. So the idea is that you can go in and I can edit a region here. If I have it up here, and then let's see in Firefox, let's see here. Actually, I think this is on Icebox here. So this, I have it, the region in both places. If I go in and edit it in one place, let's see here. It will allow me to 412. So if I go in and edit, you'll see the change occurring immediately. On the other side, hopefully. Let's see. Oh, I did the longer one, not the shorter one.
You just changed the sort of location to extend it.
Yeah, so in this case, I'm just extending the feature, but I'm syncing into the wrong place here. Problem when you're zoomed in as much as I am. So you can see that actually they're switched apart here. Oh, and this is the exon here. So I have lots of things. There we go. So you can see it changing there. And so the other neat thing about it is once I have it, I can go in and you can see here's a history of all the different changes that I've made to it. So so I can see not only what those how many things, you can actually see the structural changes to those. So if I can actually go back to that, and now we've reverted back to that newer position. Additionally, let's zoom back out again. Let's see here. Okay, so let's zoom back out again. We can do we can split. Let's grab another isoform here. So we can split these exons. Another nice feature put in by one of our developers, the ability to, if we do, so you can see that the phase, the CDS frames are colored here, so you know which one you're on. And you can also see that these are not related genes in transcript, so it actually automatically calculates the overlap, so you don't have to manually set what transcript goes on to what particular gene. I think additionally, when you, well, like I said before, you have the ability to change the type, and this is of course reversible. By default, when you drag up an element, you know, in this case, you can see where different elements are overlapping. You can they're mRNAs by default. So let's see, let's go in here. This is the different color scheming, I think. I don't know if it's exactly what you want, but it's a different view of the sequence data. Trying to think of some of the other things. You also have the ability to annotate changes to the genome as well. And those get picked up when you do export as FAST or GFS.
You can change the underlying sequence.
Yeah, reference sequence. Well, you make annotating. Yeah.
You don't literally change the genomic sequence,
right.
But you say that you believe there should be, that there is an alteration here, whether it's manual or native. But the impact is felt is registered on any transcripts from that region or the translations from that region. So if you feel like you've got a faulty base call, and then if you just corrected one base, it would match the proteome that you're comparing it to. You can go ahead and do that, and then you'll get the proteome.
Oh, that's cool, because this is a common problem. So we do we do a pack by a genome assembly, we annotate it, and then we find out there's a few little polymerase or frame shifts. We don't want to do the rebuild everything from scratch. So this can store that delta.
Yes.
And export it out later.
Yeah, it's just because a lot of these genomes that we've been working with now are, the assemblies are they're questionable. And so they really wanted that ability. What we're finding though is that when people are starting to do more populations of stuff, and you want to annotate an individual, you can use the same mechanism to say, here's what's going to be different in the proteome of this individual, because they've got a variant right here. So, and yeah, we're using the dbSNP definition of a small alteration, which is 50 base pairs are under. So you can do an insertion or deletion of up to 50.
I guess I should probably explain to people what all this stuff is. Like when you look at your JBrowse, it was just some genes, right? But Nathan's got all these different things. He's got all these different tracks, they're called. So what are some of the tracks you've got there? You've got
So thank you.
This is honeybee, by the way.
This is honeybee. So the different tracks, so these first top two are GFF3 tracks. So you have your GFF3, you run a script on it that turns it into something else.
Which is equivalent to, say, the GFF file that we got out of Procker and then put into JBrowse. It describes the common features, but you've got other feature tracks as well.
Right. So it also imports BAM and bigwig files. So these two are bigwig files up at the top, just slightly different configuration in JBrowse. I'm not sure which of these have been implemented in JBrowse, but the Apollo version in the Galaxy just wraps the exact tool that you guys are also using as well. This is a BAM file. So these are the sequence reads that come in. I don't know if you just saw me create an annotation off of those reads. And there's the sequence there for it. What other files we have? We also allow variants. Yeah?
When you're doing that, that purple lines up with the purple reading frame?
Yeah.
Okay.
And then
Sort of what you were asking.
Yeah, that's what I was asking. So if you move something that came from a different reading frame, it would show up in the same color as the reading frame.
Exactly.
Right?
Yeah, so you can see I just switched it. I just moved this one exon over, or one base pair over, and now it's lined up with a slightly
That's
different
a
one.
lot more readable than Atari 2600 being Super Nintendo.
Yeah.
The little red lines are where the edges end up.
Line up, yeah. So it's useful as you have a lot of BAM data coming in. So I'm trying to think if there's anything. So as you can see, the annotations are listed. Trying to see if we have one that has multiple scaffolds on it. So let's do.
Also, Nate, I'll just.
Yeah, go ahead.
That little exclamation point inside the circle, that means you have a non-canonical splice site there. So if it does, it's not the normal except for donor site, it gives you a little like, this is weird symbol.
No, we don't have that in bacteria.
So is it?
Well, no, but you do have different translation tables. And like you were showing switching, and that's one thing, I think, do we have that yet? I know we
Multiple different translation tables?
Yeah.
Yes,
So you can figure for the different.
we
We
do.
have that, and we also have proteins that don't translate. The amino acid doesn't match the codon. There's weird exceptions like selenocysteines and things like that.
Yeah, but it's selenocysteine, we were just talking about that the other day. Or where it's sort of, you get a stop codon read through, and instead of a stop codon, you get a selenocysteine.
Sometimes, yeah.
And then-
It's
Yeah,
all sorts of weird
We
exceptions.
have we used to, at least in Genome College, we never do that. where you
Did
can
set
say,
a read through?
Don't stop at the stop codon, it's going to be translated.
Yeah, the set read through stop. No.
Yeah, which is sort of, maybe it's not the right way to phrase it, but that's
Yeah,
effectively what it will do.
Follow use the database to store the data that's going along.
Yeah.
How does that work? The thing is, Galaxy, has the idea of basically mutable data types.
Of what?
And Galaxy data types basically mutable.
Right.
So how is this thing
It's
Working
not a
in
Galaxy data type, is it?
No. It's
so
so.
where is it basically like running the Galaxy view, but it's talking to a database of
Yeah, so it's using Docker Compose to build a Galaxy, Docker image, which is Apollo, which runs in Tomcat Java server, which runs using the Postgres database. You can use other databases, but that's the one that supports Chato, and it works well with. So that's running there. And so it's a great question, because the way we get it back out again is, refresh this here. Actually, I don't even need to do that. So what you do next is you can obviously just export it from the tool directly, but that's kind of defeats the purpose. You can click on retrieve data, and this should bring up the proper data there. So it should grab anything that's in your, I don't know what that is, that you have as a GFF3 and a FASTA. And that'll come back, which is, of course, the point of running in Galaxy, is you can bring those workflows back in. So it's going to create a GFF3 and FASTA downstream. And if someone creates a tool for re-merging those with existing GFF3.
As you can see, this
this galaxy you're running, see in the top corner, well, this is your one, but on our one, it says galaxy/GVL. You want to talk about how we have WebApollo and Postgres all set up? Is that working yet, Simon?
It is on a test server.
No. We're aiming to, we have a cloud image that runs Galaxy and a bunch of other things, and WebApollo is something that's kind of like a pre-configured plugin on there.
So I have a Docker Hub image for Apollo with the most recent
It would
version.
be nice to know before we run out.
Yeah.
Yours might be better. So
The thing to notice is it's all the data is stored in an SQL SQL database at the back end. And the schema, the table scheme for the technically honest, is called Chado. I'm not sure.
kind of. It's close to Chado, but we write out to a Chado database. So.
I wanted to say that if it is in Chado, that there's other tools besides WebApollo that can read that same database and manipulate it and view the annotations as well as this. So Artemis can do it. Artemis is a bacterial genome sort of tool that can read a Chado SQL source. The original Apollo can, I assume.
It does it the same way. So it actually stores things internally using a Berkeley DB with flat files, which writes to serialized data store. So we're using a database decided not to use a direct Chado. I'm not so sure if that was the right decision. It gives us a little more flexibility if we decide to do other things. We probably could have made it work with Chado as well.
That was just for the technical people here.
Yeah, sorry.
Boring you on that.
Yeah. So, but it is important. It triples another one that runs if you have a Chado database and you want to disseminate your data. Works well as well. So, anyway, yes.
So you mentioned that one of the benefits of having Apollo in Galaxy is that it can export data into the screen.
Yeah.
Is that the main benefit to having it in Galaxy?
Well, that's a good question because I know Eric Rashi, who did a lot of this work, used it for teaching. It makes it really easy for him to set up and build a big functional annotation workflow that comes into Apollo. You absolutely, of course, don't need to use it within there, but the big, and you can always export stuff and then upload them directly out of there. Having it run within Docker in a suite like GVL, I think makes sense. It's up to the people who end up using that product in terms of how useful it is to have it in there. It's pretty easy. I think there's additional functionality that needs to go in there. I think the biggest one is, you guys can correct me if I'm wrong, you only get campus tracks when you import data as opposed to HTML tracks in JBrowse.
I've got no idea.
Okay.
As in Canvas, as in...
you can't click and drag them up. You have to. So when we create an additional, sorry, this is kind of going a little esoteric, but when you actually drag these up, there's a certain type of track.
I don't know if it supports.
Yeah.
You seem to have a lot of different menus to what I'm saying. So
I see.
it's probably heavily customized. I don't know.
No, not too much. I don't. Yeah.
Eric's tweaked he's minimized. He's changed.
Yeah. So, that's it.
So are there any real genome curation projects that are using this?
Apollo?
Yeah.
Oh, yeah.
No, that sounds like something harsh, but I'm interested.
No. Well, I was.
Is Honeybee Apis aries or whatever it is that they're using, or is this just a demonstration at the moment?
Yes and yes. This is being actively.
You just screwed up their annotation.
Oh no.
Yeah, we did. First, we're collaborating with Chris Elphick's group at University of Missouri. She's doing honeybee and also bovine, so cattle genome. And so she works with all the people who are interested in that, and like their honeybee paper was published using their proteome and all that was published using Apollo for their annotation. And it was definitely communal. As far as other groups,
Vector-based.
Gosh.
I5K uses an older version of Apollo.
5,000 insect genome from the USDA. It's using it. They don't have anywhere near 5,000 genomes yet, but at least it's being used for 58 of them. So so far, that's what they're up to, is 58.
They have them, they're just still in their cellular form. They just haven't sequenced them.
Koala.
Let's see.
So do you find the people, the curators, do they have, how was it getting them to use it?
They like it.
Was the learning curve high? Or?
Yeah, no, well, a lot of them have experience using other annotation tools, and the fact that this one, A, is a website, you know, it's just easier.
Do it anyway.
You know, for them to get to, you don't have to install it on your laptop, it's just there. And it's because it's real time, you know, there's somebody in, where are they? They're in Panama and in, I forget, Colombia or someplace like that, using it and the Polyphonians, the butterfly.
Yep.
Hardwood group, the plant group,
They
yeah.
can keep working, you know, seeing the same screen at the same time and work together.
Eric Rashy's phage group, that'd be another one.
So
Yeah.
The virus.
generally, groups find it much easier than Web Apollo was. The Web Apollo, there's still some functionality that it has that the web version, I mean, the desktop version, I'm sorry. The desktop version is maybe had more functionality, but was harder to install and use. And, you know, hooking, the biggest problem most groups face is hooking it up to an upstream pipeline. You have to run something like Coca.
Yeah.
You have to have that. And one nice thing maybe with Galaxy is that if they decide that they want to add another track, they can just go run another analysis and slip it in that way. Which would be a lot more convenient for people. It really is the major bottleneck is like, are you going to get data to it?
If you can get anything into GFF format, you can then pull it into Web Apollo.
Yeah, and I know that Eric and Bjorn, and I don't know who else have gotten it, so you can add BAM, or at least Bigwig tracks, and I think a few other ones. So it's not.
And VCF as well.
Yeah, VCF as well.
VCF.
One
Okay.
of the things I see people shifting to is they'll be interested in a particular protein family, like, I don't know, maybe something like factory receptors or something like that. And they'll just want to look at a bunch of organisms and see all of the olfactory receptors, and nobody else's sequence in detail, you know, those olfactory receptors. So they want to align them and then go in and see what does that do. So I could see it being used that way possibly. For people who are interested in families.
Yep.
So it's 2 o'clock now. The tube technically goes to 3 o'clock. If anyone, so we've got a few options. We can just, if you want to finish now and go, that's fine. The tube's technically over. If you've got any questions, of course, ask them. But you can leave now if you like. It's not a problem. Have a break. You probably earned it. Some of you may have noticed that on the website where these tubes are that you follow any instructions, on the menu there's an extra one at the bottom called variant calling, which is about finding SNPs and finding out their effect, the consequences of these SNPs on a genome. I'm happy to stay here if anyone wants to work through that sheet. If anyone's interested in SNP calling, it's not official, it's not an official tutorial here, but you could probably do it in an hour easily. So I'm happy to stay behind if you want to do that, but otherwise you can go. I just wanted to thank you, Nathan. I think we've got a good idea of what's going on now.
Yeah, I hope so.
Thanks for sort of coming in at last minute and contributing to this session. So yeah, you can stay, do variant calling, stay, ask questions, or go, have a break. Enjoy the sun.